Kangaroo Island's koala population might seem like a conservation triumph at first glance, but beneath the surface lies a hidden genetic dilemma that could threaten their future survival. This is not just a story about numbers. It's about the fragile genetic health of an isolated population and why intervention is urgently needed. But here's where it gets controversial. Should we interfere with nature by introducing new koalas from the mainland, or let this island population face the consequences of its genetic isolation? Researchers from Flinders University have uncovered compelling evidence that Kangaroo Island's koalas, despite their relatively large and disease-free population, are caught in what they call a genetic trap. This means that because the population began with a very small number of individuals, their genetic diversity is alarmingly low, which could make them vulnerable to future threats such as disease outbreaks or climate change. Using advanced whole genome sequencing techniques, the team compared the DNA of koalas from Kangaroo Island with those from mainland populations in Victoria and Queensland. The results were striking. Although Kangaroo Island's koalas are abundant, their genetic variation is significantly reduced, and they show much higher levels of inbreeding compared to their mainland cousins. This genetic bottleneck traces back to the 1920s, when fewer than 20 koalas were relocated from Victoria to Kangaroo Island. This small founding group came from an already limited population, which means the island koalas inherited a narrow genetic base from the start. Over time, this has led to long stretches of identical DNA, known as runs of homozygosity, being passed down through generations, a clear sign of inbreeding. Why does this matter? Inbreeding increases the likelihood that harmful genetic mutations will be expressed, potentially causing fertility issues and developmental abnormalities. In fact, sun-captured koalas on the island have already shown signs of such problems. While these koalas have so far resisted major infectious diseases like chlamydia and koala retrovirus, their limited genetic toolkit could leave them defenseless against future health challenges. Meanwhile, koalas along Australia's east coast are officially classified as endangered, with populations shrinking due to habitat destruction, bushfires, disease, and road development. Kangaroo Island's koalas have become a beacon of hope for the species, especially after the devastating 2019 to 2020 bushfires that destroyed over half the island's habitat. Yet, as Dr. Katie Gates, the lead author of the study, points out, while the population size is a conservation success, the genetic health tells a different story. Their low diversity and high inbreeding mean they may struggle to adapt to new threats. The researchers recommend a proactive approach known as genetic rescue. This involves carefully introducing koalas from genetically diverse mainland populations to Kangaroo Island to boost genetic variation and reduce inbreeding risks. Senior author Professor Luciano Bejarege emphasizes Kangaroo Island has the potential to remain a vital refuge for koalas. But without genetic management, this sanctuary could turn into a genetic dead end. Continuous genomic monitoring is essential for species managed in isolation. This study also raises a broader question for wildlife conservation. While islands and fenced reserves can offer immediate protection from external threats, they are not foolproof. Without strategies to maintain or enhance genetic diversity, these protected populations risk becoming genetic traps, undermining long-term survival. So, what do you think? Should conservationists intervene by mixing populations to preserve genetic health? Or should they respect the natural course of isolated populations, even if it means risking their decline? This debate touches on the core of conservation ethics and strategies. Your thoughts could spark an important conversation. The full research article, titled Conservation Arcs, Genomic Erosion and Inbreeding in an Abundant Island Population of Koalas, was published in Molecular Ecology, 2025, by K. Gates, J. Sandoval Castillo, J. E. Beeman, K. Burke da Silva, R. Saltrue, K. Belov, C. J. Hogg, C. J. A. Bradshaw, and L. B. Beharagrai. It is available at https://doi10.111/MEC.70097. This work was supported by the Australian Research Council, LP 210100450. The authors respectfully acknowledge the traditional owners of the lands where data was collected and analyzed, including the Ngarrindjeri, Narunga, Kaurna, and Paramank peoples of South Australia. Logistic support was provided by Flinders University and the University of Sydney. All data collection complied with Australian regulations and was conducted under Flinders University Animal Ethical Approval AEC BIOL 5591-15.